Hey there, I am Maruf Said, a microbiologist and a science educator. Science is my passion and I'm here to share it with you. Microbiology topic on biochemical test, Leucine Aminopeptidase test. But before starting this video, don't forget to like this video, subscribe to my channel and press the bell icon button. Let's start with the table of content. A brief introduction about lab test, its objectives, principle, requirements, procedure, result and interpretation, quality control, precautions, applications, and lastly limitations. Let's start with a brief introduction of lab test. Catalase negative gram-positive cocci bacteria are identified and differentiated using the lab test. It is significantly utilized to differentiate between Aerococcus and Leuconostoc species and Streptococcus, Enterococcus, Pediococcus, and Lactococcus species. Leucine residues from the N-terminus of proteins and peptides are hydrolyzed by leucine aminopeptidases, which are metallopeptidases. They are peptidase family proteolytic enzymes that assist in bacterial pathogenesis as well as protein metabolism for bacterial survival. Leucine aminopeptidases, however, are not synthesized by all bacteria. Therefore, detecting lab production is a crucial method for classifying bacteria. In the microbiology lab, the lab test is employed as a biochemical test to distinguish between groups of bacteria that produce leucine aminopeptidase and those that does not. So what is the objective of the lab test? To evaluate bacteria's capacity to produce leucine aminopeptidases enzymes. To distinguish and presumably identify gram-positive cocci bacteria that lack catalase. Let's learn the principle behind lab test. Leucine p-naphthalinamide compounds in the lab disk are hydrolyzed by leucine aminopeptidase enzymes produced by bacteria. Leucine and free beta-naphthalinamide are produced by the lab's enzymes hydrolysis of leucine p-naphthalinamide. In the presence of cinnamaldehyde reagent N,N-dimethylaminocinnamaldehyde, the free beta-naphthalinamide reacts to produce a brilliant pink or cherry red shift base, which indicates the lab test was successful. Let's see the requirements for lab test. The reagents are lab disk impregnated with leucine p-naphthalinamide, and next lab reagent 0.01% p-NN-dimethylaminocinnamaldehyde is used as a lab reagent. The PYR reagent and lab reagents are the same. Composition of lab reagent: NN-dimethylaminocinnamaldehyde is 1.0 grams. Then concentrated hydrochloric acid HCl 10 ml. These all measurement is for 990 ml of distilled water. Preparation of PYR reagent. With continual stirring, slowly add 10 ml of concentrated HCl to 990 ml of distilled water along with 1 gram of NN-dimethylaminocinnamaldehyde. Make sure to thoroughly combine so that NN-dimethylaminocinnamaldehyde dissolves. And lastly, store in a dark area between 2 to 8 degrees Celsius. Equipment needed are Petri plates, Bunsen burner, inoculating loop, forceps, PPE, and other laboratory materials. Test organisms, sample bacteria. Positive control will be Enterococcus faecalis ATCC 29212, and negative control will be Aerococcus viridans ATCC 11563. Before going on to the procedure, don't forget to subscribe. The procedure of lab test. Utilizing forceps, insert an lab disc into a sterile petri dish. Use sterile distilled water to wet the disc. Using a sterile inoculating loop, pick up 1 to 2 loops full of well-isolated sample bacteria and rub them onto the lab disc. Incubate the plate aerobically for 5 minutes at room temperature. Add 1 to 2 drops of lab reagent over the seeded lab disc. After the reagent has been added for 2 minutes, watch for a change in color. Next is result and interpretation of lab test. After 2 minutes of adding the lab reagents, the disc should develop a bright pink, cherry red or reddish purple color. This indicates a positive response. Lab test is positive. Pink color development denotes a mild response and is seen favorably. After the addition of the lab reagent, no color change or formation of yellow or orange color indicates a negative reaction. Lab test is negative. Bacteria which are lab test positive are Streptococcus species, Enterococcus species, Lactococcus species, and Pediococcus species. Negative lab test bacteria are Leuconostoc species, Aerococcus species, Tetraginacoccus species, etc. Some Aerococcus species give variable results. Aerococcus urinae may give positive results. Next is quality control. Enterococcus faecalis ATCC29212 results in the formation of bright pink red color over the disc within 1 to 2 minutes of the addition of the lab reagent. Aerococcus viridans ATCC11563 doesn't form pink or red color, doesn't change color after the addition of the lab reagent. Let's see what are the precautions we must take. Keep the lab disc and reagent in the frigid, dark environment. Just lightly moist the lab disc with distilled water rather than completely submerging it. Rub the disc with a heavy inoculum that has been transferred. A faint pink color may develop or falsely show up as negative when there is insufficient inoculum used. If you have reached till the end, don't forget to subscribe. Application of lab test. Detailed classification and identification of gram-positive, catalyst-negative cocci. Limitations of lab test. Catalyst-negative, gram-positive cocci bacteria are the only ones that can be used. False negative results might be caused by insufficient inoculum. It requires additional biochemical tests to confirm the identification of sample bacteria, because it is insufficient for the complete identification of bacteria. So that's it for today. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. Thank you so much for watching till the end.